Okay. First thing to say, by the way, everyone, is just that this session is being recorded. So if you do ask any questions or if you have your cameras on, that will be on the recording. If everyone could keep their microphones muted unless they're asking a question, that would be great too. So we have 3 panelists with us today, and I'm just going to introduce them quickly, and then they'll, I'm sure, introduce themselves in a much better way than I do. And then I will hand over to Sayantani, who will start with the questions. So the first of our panelists is Dr. Ben Collins. He completed his PhD in the University College Dublin and then stayed there for a while as a postdoc, doing quantitative proteomics, looking at method development and targeted protein measurements. He then moved to ETH Zurich, did another postdoc there on quantitative proteomics to get more looking at interactions and using data independent acquisition methods of mass spec, so DIA swath, etc., which many of us will have heard about. And then in 2016, Ben became a group leader at ETH and started his own lab, applying these methods that he'd been using to study host pathogen interactions, particularly of Mycobacterium tuberculosis. And now Ben has moved to Queen's University in Belfast, and that's where he continues his work in his lab. So we're looking forward to hearing all of Ben's opinions and tips, hopefully. Our second panelist is Dr. Ling Hao, who undoubtedly had a very different experience to Ben. She did her undergrad in China before moving over to the States to do a PhD at the University of Wisconsin-Madison. And this was in pharmaceutical sciences. And then Ling took a postdoc position at the National Institute of Health, where she's done multiomics, looking at metabolomics and proteomics, particularly in neuroscience applications. And Ling's now an assistant professor of chemistry at George Washington University, where she applies these multiomics methods within her group to study lysosomal and mitochondrial components of brain disease. So we're looking forward to hearing Ling's opinions too. And our final panelist is Dr. Leonard Martins, who is on the bioinformatics and computational side of proteomics. He did his PhD at Ghent University and then became a software developer for EMBL. And many of you will have heard of PRIDE. So Leonard was actually responsible for the first version of PRIDE, the proteomics data repository that most of us use. And he became the group coordinator for the PRIDE project at EMBL before moving back to Ghent University as a professor in systems biology. And he's a group leader there now and continues his work. So we're looking forward to having 3 very different opinions and experiences in this panel and hearing about how you 3 have found starting a lab. So I think each of our panelists should have a few words prepared, 5 minutes, about their background and their experiences. So I'll just go in order that I introduced. If you wouldn't mind starting for us, Ben.
Great, thanks a lot, Charlotte. Let me get my screens in order here. I hope you're seeing my slides now.
Yeah.
Yeah, so thanks a lot. I want to thank a lot the HUPO ECR and initiative and the YPIC for the opportunity to talk to you today. So yeah, I thought maybe by way of introduction, I would say something about how I got here, my career path. So the early years, I think, are kind of fairly standard with some bachelor, master, PhD, and so on. Also a few years kind of out in, let me get the laser pointer, out in industry. A postdoc sponsored by a vendor, which was a helpful thing. And then I think the major turning point was moving to ETH Zurich to be a postdoc in Rudy Ebersold's group there for about 5 years. So this was obviously an important opportunity for me and very formative. You know, at that point, I had to make the decision after this 5 years postdoc whether I was going to leave or try to stay. And I took the decision, which is maybe somewhat of an atypical career path, to try and stay at that institute for a while. So I got an Ambizione, so-called Ambizione fellowship, which was this sort of career transition award. And that allowed me to set up a little group, which, if I'm honest, was really kind of stapled onto the side of the Ebersold group, but allowed me to have some kind of independent research And I think this is a possibility for people, but not the normal path. And maybe I'll go a bit more into detail on that. And now I'm at, as Charlotte mentioned, at Queen's University in Belfast as a reader, which nobody knows what this means. It's basically similar to associate professor, but they use this kind of old British system where they don't call you a professor until you're a full professor, much to many people's chagrin. So yeah, I think maybe for the purpose of today's discussion, I'm going to zoom in a little bit on this kind of later period. So there's a sense in which I kind of did two lab startups, sort of first in Zurich and then now more recently in Queen's. But the first one was in a way very easy, right? So I had this career transition award, which allowed me to have kind of a student and a technician and some other co-supervision of some other students. I had my own kind of money. I was paying my own salary. So I was considered in some way an independent group leader. But I still had very kind of strong ties to the Aebersold group and was also sort of somewhat functioning as kind of a senior scientist in that group as well, and was involved in a lot of those kinds of activities. So this was great in a way, as a way to get started, because I didn't need to worry about instrumentation and equipment and, a sort of critical mass of talented people. All the instrumentation and things that I needed were there. There was tons of experienced people around, to help when things went wrong. And so in a way, to start your own research program, I thought that this was a very nice environment, actually. Excuse me. I think it turned out to be actually scientifically fairly productive for these reasons. I mean, we were able, I spent, sort of 3.5 years in this position only, but we were able to do some interesting things during that time. And I think that's really attributed to the fact that I didn't have to do this kind of startup stuff. When it became a lot more real is when I moved in 2019 to this lab in Belfast. And there it was, I had a higher level position, but I had to start basically completely from scratch, completely from, buying the first vial of trypsin to all the rest of it. And this was rather scary and took a long time to do. From a practical perspective, there was some money for instrumentation associated with this position, and also a first postdoc and some project funds. So I had, somewhere to start. So we could get going, but then actually we had some rather bad timing. About 6 months after I moved there, we had the whole COVID pandemic lockdown and so on, and this was a real killer, and it was actually really just a very difficult time. Of course, it was a difficult time for everybody, but just this moment, if you were trying to kind of establish things and get set up, it was just very bad timing. And so, yeah, it's really only, I would say, the last 6 months or so that I feel like the group starts to kind of produce, productive work. Of course, we tried to do as much as we could during that time, but it was very much restricted in terms of hiring and lab access and many other things. And so, yeah, to summarize this part, I mean, I basically started up 2 labs. The first one was easy and it was a lot of fun. The second one was harder, but kind of still fun. So having this first lab, attached to the side of the Aebersold group was very convenient, but it in a way delayed the pain of setting up my own lab. And I'm not sure if I would completely recommend this, but it is one way to do it, not the most typical career path. And so I thought it might be interesting to mention, a bit what our interests of the lab are. And this is not just to kind of, so I'm trying to promote myself, but I think it's kind of important in some way that you have some identity, that you're able to summarize to some extent what your research interests are. And so I kind of put this into 3 bins. Probably my strongest part of my background is in these kind of quantitative proteomics technologies, this DIA swath type of stuff, and we spent many years working on that, and we continue to try and work on that. I also spent a lot of effort on protein complexes and interactions, and now more recently I'm trying to apply some of these kind of underpinning technologies to problems in the area of infection and immunity, and we have some good collaborators here locally on this. So the reason why I bring this up is to say, like, I think it's useful to have some encapsulation of what your interests are and where you intend to go. Have some idea of this in your mind. And for me, my strength kind of came from the technical part, but I try to progress into this more biology part. And to some extent, for the kind of more technically oriented people out there, I think that these days it's a little bit hard to survive on technology alone. That's not entirely true, but it does help if you have some biology interest where you can kind of pitch at certain other kinds of grants. So I think, going back a few years, it was possible to be a purely technology driven person, but I think that's kind of harder to do at the moment. As I was kind of thinking about what to say here, I thought I might mention what I think is probably the biggest challenge when transitioning from like sort of postdoc level to group leader, PI level. And for me, it was this kind of idea of having your time and attention fragmented into like, 100 different directions. And in principle, if you're a postdoc usually, or a PhD student, usually your job is to spend majority of your time on research and be kind of focused deeply on this. At least this is the idea. But as soon as you get to the kind of group leader stage, then you get pulled in a million different directions, like this poor guy here. And some of this can be fun, but some of it can be just very difficult and things that you don't necessarily want to do. So I feel like this is the thing that changes the most when you move to being a kind of a lab head. And I think you need to be prepared for this. And I think I was prepared for this mentally, but nevertheless, it was still a shock. And this is, if I'm honest, the part of my position that I probably struggle with the most. Then of course, we're going to get into these things more later on in the discussion, but I thought I would also, try to, if I could encapsulate it in a nutshell, some piece of advice, I think it would be something along these lines, which would be to be able to say no, first of all, because of this problem, you get pulled in a lot of directions and you need to be able to prioritize. And that actually means kind of saying no to people at some point. You can't always do this, but you feel as a young group leader compelled to help people and do things when people ask you to do them. And you need a little bit to find the courage, let's say, to resist that at some moment, or the discipline. The other one would be to kind of try and focus on high level goals and always keep this in the back of your mind, what you really are trying to achieve. And for me, it's kind of encapsulated here, right? So these are the directions that I want to go in. And so I'm trying to always, when I'm, taking some decision, asking the question, like, am I doing something in service of these goals here? And then the corollary of this on the third part is to try not to sweat the small stuff. This is, of course, easy to say and hard to do, but if you're going to be worried and sweating about things, you may as well be worried and sweating about the sort of high level goals that you're interested in. And so these are my so-called pearls of wisdom to kick things off. So thanks a lot for your attention, and I'm looking forward to this to the discussion. Thanks, Charlotte.
Thank you very much, Ben. And if you could
turn off the
screen
share.
sharing, please. And I think we will go straight to our second panelist, Ling, if you could.
Sure. Let me share my screen.
Perfect, thank you.
Yeah, so I think I have the exactly same kind of flow as Ben did. So I'm going to talk about like the lab and myself, and then some of the tips. So my lab started in 2019. So now I have 3 PhD students and 3 undergraduate students in the lab. And we are located in Washington, DC, George Washington University in Department of Chemistry. So just a little bit of myself. Well, I'm from China. I did my bachelor degree and it's in chemistry. So I'm trained to be a chemist. Although I joined a pharmaceutical science department for my PhD, it's also kind of very chemistry heavy. We're using it's a mass spectrometry lab in Dr. Lin Jun Li's lab. And we have both pharmaceutical sciences students and students in chemistry departments mixed together in the lab. So it's very chemistry heavy. And that's where I realized that that's where I gained all my like mass spectrometry expertise, metabolomics, proteomics, and peptidomics. And then when I realized that some of the studies is really, we ended at kind of the volcano plot stage. We find a couple biomarkers, then end of story, and we moved on to another one. So I really wanted to get more learnings in the biology and especially neuroscience. So I joined 2 actually hardcore cell biology labs, which I can talk more about it. So this is something it's totally different world. And I think it's really beneficial and I learned how to communicate with a biologist, and we really speak different languages. It's a joint mentorship. So I was really doing 2 postdocs at the same time, because there are 2 different labs that does very different things, that they both want to do some mass spectrometry, but they also have their own research projects. So I was doing proteomics at the same time of learning the biology. Then I started in Washington, George Washington, really actually a couple miles away from NIH. Started my own lab in Department of Chemistry in 2019. So that's kind of my trajectory. Because my trainings in both mass spectrometry and neurobiology and cell biology, now I wanted to do both and combining them to study the neurodegenerative diseases. So my lab currently have, like I mentioned, 3 PhD students and 3 undergraduate students. I think this is one thing that we, when you are looking for jobs and also when you're starting your lab, you have to think about, like, is your lab going to be a student heavy lab or a postdoc heavy lab? One thing is depending on your priorities, another is depending on the institutions you are at. So normally at a chemistry department, you would have more students. At a school of medicine or a national lab or government lab, you would have more postdoc. And so you can kind of have to decide on who you wanted to work with, and they are very different. So for working with young students, you really have to, I think I gained a lot of patience over the past 3 years and also tailor the projects to the students' needs. So in terms of starting my lab, I think I'm just going to provide one thing per category. As Ben mentioned, there's so many things we have to deal with, and we were trained to be a mass spectrometrist, but we were never trained to manage the lab. So one thing I found really helped me along the way is when I was at PhD students or when I was a postdoc, I often think, like, what would I do if I was the PI? So I asked myself many times of questions like that. So that really helped me learn from the supervisors when I was a student, and also ask them questions like, why do you do things like this? Why do you do it in this way? So it really helps me, I think, along the way of when I was kind of at the end. I wasn't ready to start my lab actually, because I had a pretty short postdoc training. I've only had 2 years of postdoc training. So I was just kind of trying. I think my graduate advisor was encouraging me to try it for 1 year. And I get this, so then it happens to be my job. I really liked it. But although I wasn't kind of sort of totally trained up, I think, as a neurobiologist, but I'm mentally prepared to start my lab because I've thought about all the different things that are going to happen in my lab, and I sort of have an idea of what kind of lab I wanted to build. So in terms of job search, you wanted to decide there's different kinds of positions, and they all have their pros and cons. For universities, if you are in a chemistry or biology or biochemistry department, you have some teaching duties. So whether you like teaching, that's one question to ask yourself. If you start in the national lab, usually you have to sponsor your whole salary with a grant. So you have a lot of grant pressure, but you don't have to teach normally. And you're mostly working with postdocs. I think Scripps have some graduate students, but most the national labs and government labs are similar, that you don't have a lot of graduate students. Where government lab, especially at US, like NIH and NIST and FDR, they don't have to apply for grant. So they can apply for some foundation grant, but most of their money is hard money sponsored by the government. So you focus on the high reward, high risk, high reward research. So they're very different. So that's something you wanted to think. Do you like writing? Do you like writing grant? Do you like teaching? And for me, I actually like all the different aspects of it. And I kind of I really like research, but I also like all the different things I can do. So that's also actually one of the reasons I think I really enjoyed my current job, because every day is a different day. And then in terms of purchasing, so this is where I think we're going to start more about that in the as we discussed. But I had to buy my mass spectrometry with my startup. So that was the one thing I think I had to decide. And this I talk about, like, whether postdoc or graduate students and teaching duties, like you can't, I think you can spend a lot of time in teaching and making it perfect, but balancing the time between teaching and mentoring students and writing is also a lot of things. I think I'm gonna go quickly here so we can go to the next speaker. So just about the research direction. So we I was thinking when I started my lab, I wanted to build both the mass spectrometry side and the neuroscience side. But we started with the mass spectrometry because that's where I'm mostly familiar with. And also because of pandemic, hiring and purchasing was pretty challenging. So there are three main directions in my lab. One is mass spec technology advancement. We develop proteomics, mainly proximal labeling and protein turnover to study organelle dynamics, lysosome mitochondrial. And we also use proteomics and metabolomics to study to try to discover biomarkers and combining them in the same pathways to understand the disease. And we try to understand disease mechanism, in this case, neurodegeneration, using the iPSC-derived neuron platforms. So that's the main platform my lab work on, but I didn't start culturing neurons until last year, actually, because of all the postpones. But we did have all the mass spec platform set up at the beginning. So that's just some pictures of my students and the lab space. And that's it.
Perfect. Thank you so much, Ling. And Leonard, if you could share the screen, thank you.
Okay, so you should see my screen, and I hope you don't have too much background noise because I'm actually in the airport, as I usually am. And so if there's a lot of background noise, you know where it comes from. So I have also some notes on starting my own lab, but to be quite honest, I don't remember that much anymore about the period because it was a bit of a daze. Let me try and convince you as to why it was a daze. So this is a picture of me. As you can see, I was a lot younger then. This was me right after my master's degree. And yes, I know I don't look the same anymore. But I then actually worked for a while outside of academia. So I worked as a business software developer, putting in some hard work there. And I'm going to make some comparisons with industry because I think that might be relevant as well. But I came back and did my PhD, and then I started to look a little bit worse. And then I went for a postdoc, and then I got a lot worse. And then I was a starting PI, and I died somewhere along the way, I think. But you know, things get good once you become a senior full professor. You can then do this to other people, essentially. So of course, this is a bit facetious, but it is kind of the same thing that Ben mentioned, is that the workload does increase as you go through your career, and you have to be very prepared for that. It will never get better, regardless of what you believe. So if we look at the winding path of my career, actually, it's not that atypical. It's just the orange bit where I worked as a software developer. That's slightly different because I stepped out of academia. But fundamentally, I did my master's degree, or licentiate thesis, as we called it at the time. Then did my PhD, and then did a Marie Curie fellowship at EBI to start Pride, as Charlotte already helpfully mentioned. Finished my PhD in Ghent, went back to EBI to run the Pride database. And that was essentially my first building of an own group. What was special about that was that there were already a few software developers there that I was supposed to take charge of, and everybody was older and more experienced in what they did than I did, because I was fresh out of my PhD. And that was an interesting experience, actually, to guide these people and to try and earn their respect at the same time, which is non-trivial. And then in the end, I returned to Ghent, where I'm now a senior full professor, a group leader, and associate director, and I'm still a visiting scientist at EBI. And so I rebuilt my group completely from scratch at Ghent. So I've done it twice, and in slightly different circumstances, so it would be fun to discuss it in more detail during the discussion. I also just wanted to throw out some numbers, which I have for Flanders, the region where I work. They may not apply across the world, but I think they're probably reasonably similar, just to show you what it looks like, a career path in academia, when you look at the honest to God, clean, naked numbers. What you see is that lots of people get their master's thesis, and then a huge number of people go somehow into industry, go into a quote unquote regular job. And then some people go into a PhD. In Flanders, there's quite a few people going into PhDs because we have quite a lot of PhD funding from the government. That may vary. And then a lot of people after the PhD go to industry again. And then some of them make it into postdoc. And then a lot of postdocs at some point jump to industry. And then very, very few of them make it to professor. And then some professors actually leave and go to industry because they don't ultimately make the tenure, or because they get hired away by industry. Now, industry does supply some people back. Like, for instance, myself, I came back from industry, but these are the rare occasions usually. In Flanders, there's an interesting chart that you can look at for the past so many years. And what you can see is that the number of PhDs has been growing quite rapidly. The number of postdocs has been growing quite rapidly, but the number of professors does not really follow that curve, which means that the competition gets harder. So that's a very hard truth in the region where I'm from. Back when I became a professor in 2009, the situation was better than it is today. Fundamentally, if you're a starting PhD student. And when I got my PhD in 2006, the situation was even better. And when I got my master's in 2000, the situation was better yet. The other thing you see is this is how many years after PhD, or when essentially you got your PhD, and how many of those PhDs that make it to professor. And fundamentally, it means that the longer you wait, the harder it gets to become a professor. So you have to, at some point, try to get advanced early enough in your career. Okay, that's the Flanders thing, and it essentially just summarizes it's hard. It's not that easy. So some key points for success, and I want to compare academia with industry here for a moment because I have some experience with both. In academia, it's vitally important that you become known for something. So people need to associate you with an advance of some sort. So you're the person who does X. That is really quite important because that's the kind of people that will be hired because you can do X for them or because they want X and they open a position for you. Obviously, for that, you need to publish a lot and preferably with high impact. And then we can have a big discussion about whether or not impact is a meaningful metric for anything, but that's the name of the game. International experience is something that is extremely highly valued typically. So they want people who have been around, that have seen other labs, more than one. And then obviously at some point you will have to obtain lots of funding to keep everything alive in your group and to stay competitive. In industry, it's very different in the sense that you need to understand how a business operates, which is something that they don't really teach you in school, especially not if you're in the life sciences. So I have people in my group who went for MBAs, so because they wanted to know how business operates. You need to build up the tech skills that are in demand. I have computational people, they're very much in demand, for instance. You have to build up your people skills because companies really value those, much more than academia, where nobody seems to care very much about your people skills with all due results. And then finally, companies really like it when you have leadership experience, especially if you come in with a PhD or a few years of postdoc experience, because they expect you to enter at middle management level. So you're supposed to be able to handle people in your team. In both cases, it really helps to build a strong network. So to know a lot of people and to have a lot of people know you. It always helps to be self-motivated, obviously, but I guess everybody is on this call. Very importantly, I've learned that it's critical to persevere because you will have really bad moments at any point in your career. They just get worse. And then just to believe in yourself and keep going is unfortunately a cliche, but a very useful one. And then finally, we're mostly an analytics-like field. So you really want to learn some structure and some attention to detail because everybody who's ever going to evaluate you is probably going to be a nitpicker in some degree. So that really helps. Now, an academic career hinges on smart choices, I think, on hard work, on raw ambition, and a lot of luck. One of the things you can do is find yourself good promoters. And we've heard two examples of people, two previous speakers mentioned their promoters explicitly and the impact that they had, because they really do change the way you provide your output and your impact. And they can really help you with your international recognition and your network. EBI for me was a place where I built a network like crazy, for instance. Very often you need to look beyond your local area or country unless you're in a really big country, because typically there are many more opportunities outside of the place where you are right now than the place where you could potentially go. And we're academics, we are supposed to be highly adaptable people. We should be able to go anywhere and somehow fit in. Very importantly for yourself is to build an actual vision to make a difference. So you have to figure out where your field will be going in the next 10 years, and you need to start working on what you think will be relevant in 10 years today. And then you have to try and make people take notice. If you do that, that's a surefire recipe to get somewhere in your career. It's not trivial, but it's definitely something to think about. Maybe briefly, just to contrast what I like about academia after having been in industry, is that in academia, I am my own boss and I am solely responsible for my group. So nobody tells me what to do. The downside is that everything is my fault if it fails. So everything is on my shoulders, and you shouldn't mind being the lone boss. And that is actually less fun than it sounds because you have very few people to talk to. And in companies, this is better because you tend to be with multiple managers, and at least you can share the blame and you can share the failures. You also have to share the success, but that's usually easy. one of the cool things about academia, and I cannot stress this enough, is I get to work with super smart people who are all highly motivated. And you should turn that into your strength. You should never be afraid of hiring people who are smarter than you. If you start a lab, find the smartest people out there, the people who impress you, the people who intimidate you because they're so very good, and hire these people. Get them to come to your lab. The nice thing is that as a result, you need to stay on your toes all the time. And so it's important to keep up with all these smart people around you. Really, really nice about academia, because we have to say nice things too, is you get to see people grow and flourish. And then, of course, these people that you've trained and have become really, really good at what they do, they go on and go do amazing things elsewhere, and you start again and see these people leave and start again and see these people leave. So after a few years, you start to feel the grind of this, but it's what we do. It's we train people, and we hope that these people make really brilliant careers. And then finally, I think I've managed to make some really amazing, completely crazy friends all over the world in my field, which is cool because they like the kind of stuff that I like, and they're into the same things that I'm into, because we're all in the same science. And I probably also made some amazing, crazy enemies, but hey, that's fun too, right? So finally, I'm going to advertise the HUPO ECR here for a moment. This is not rehearsed, so Charlotte does not know this is coming. But I think one of the cool things that you can do to advance your career is to get your hands dirty and to join an organization like the HUPO ECR and help organize things like Charlotte is doing now. It helps you meet new people. It helps you learn new things, new skills, and then you can make an impression on a lot of people who are established who will say, hey, this person is doing a fantastic job. They're really good at this. This is definitely good. So taking on an active role in such an organization is a really great boost for your career. It provides an excellent networking opportunity. It gets you noticed, at least when you put in the hard work. So if you do nothing there, then of course nobody will notice. But you also get to acquire a broad set of new skills and competences, and you can develop some self-confidence. And in fact, you can organize these things, because organizing a lab, as we have heard several times, is quite a challenge. So, don't think about it and join the HUPO ECR today and make a difference. Well, anyway, these are the people in my lab. These are the super smart people that are willing to work for me, and I'm super, super happy to have all of them. They really make all the difference. I could tell you amazing stories about every single one of them, and they will all go very far. But in 4 years, they will probably not work for me anymore. That's the only downside. And finally, I have some stupid PhD comic things that I think are relevant and funny, and I will leave them here for a small moment while we mosey in into the questions. Thanks.
Thank you very much for that. If you stop sharing, I think I will hand over to Sayantani, who's going to start the panel discussion and the questions.
Thank you, Charlotte, and thank you to all the panelists for the really interesting insights. So to start off with our first question, what can ECRs start doing to prepare themselves and be in good stance for starting their own lab in the future? Would you like to start, Ben?
Yeah, sure. I mean, I think something Leonard mentioned is very important. So he suggested everyone should join this ECR initiative as a means to build the network and so on. And of course, that's not the only thing you can do, but something that I did during that time was to do a lot of, for example, teaching of workshops and week-long courses. And I think that had a similar effect to the kind of thing that Leonard was mentioning. It meant I got exposed to a lot of people and grew a network, right? So there are things that you can do to kind of to grow your network. And that sort of helped me a lot. I think that trying to spend some time in a good lab, in a high level lab, of course, there are many good labs, but having experience in a high level lab is really seems to open some doors, both from the perspective that you get to do high level work, presumably, but also you get to know people, and this is what Leonard mentioned in terms of promoters. So if you can have access to some high level labs, even if it's only for a visit or better for a postdoc, then I think this just makes a huge difference, whether for right or for wrong. Yeah, I mean, there are other kind of more pragmatic things. Try to get involved in some grant writing and so on. This certainly helps. Learn a bit how to write and how to present your ideas, these kinds of things. But probably at the end of the day, the research and publication record and so on is what's going to make the biggest difference. So that should be the core activity. The previous 4 or 5 are should be in some sense peripheral to this.
Very interesting. Ling, do you want to carry
Sure.
on to us.
Yeah, I totally agree with Ben. I think there's all the other things that you wanted to get experience in, but there's the core of your publication record or grant writing record that really gonna decide where you're gonna how long and how far you can go in terms of job search. I'm gonna focus more on like the happiness inside. I think we all wanted to be happy. I think that's the true, I'd say, meaning. I mean, we wanted to save the world as a scientist, but as a person, we want to be happy. So I think as a PhD or a postdoc, you wanted to start thinking about what you like and what you don't like. So first you have to make a choice of either industry or academic. And sometimes it's very hard if it is hard to figure out what you really like about this, it's much easier to figure out what you hate about. So if you really hate about writing grants, then that's something you wanted to seriously consider whether you want to be in academia. I mean, there is a way you can get around with it, right? Joining NIH or government labs where you don't have to write a grant, but you limit yourself to those limited opportunities. So I think figuring out what you hate is something that you can do as a PhD and postdoc. And you wouldn't know whether you like or hate it until you try it. So you think about all the aspects of being a PI, teaching, mentoring, writing grants and papers, and leading a team, managing the team. All of those, you can sort of have a little bit of experience when you are a trainee and try them and see whether you like them. And then you will be more happier, much happier when you are really starting your lab. Yeah.
That's a very interesting perspective. And Leonard, would you like to carry on from there?
Yeah, some very, very useful things have been said already. Maybe from the point of view of having been on a lot of committees that appoint new PIs, I can tell you what people look for in these committees. First thing is, what does this person bring? What is the research? What is the focus? What is the vision? And will that fit? Is that something that we are looking for? So you need to have a well-defined academic CV in terms of the papers that you have, not just the high impact papers, but are these high impact papers driven by you? So are you the first author, or are you somewhere in the middle? And second, do they have a common theme? And can you elaborate a bit on that common theme? Because typically you have to present a research plan of some form when you apply. So where is this coming from? Where is it going from? Does this make sense? So you need to have that vision. The second thing that people look at is, to what extent have you already managed to get any funding yet? Because if you have received any funding, and these can be personal fellowships, these can be travel grants, whatever you can write down, or have you co-written anything with anyone where you were not allowed to be the PI, maybe because you didn't have the seniority, but at least you can say, hey, I co-wrote that grant. These kind of things are very important. And then the final thing is leadership and people management. So have you supervised any students? So as a PhD student, try to supervise master students. If you've done that quite often, that looks a lot better than somebody who has none. If you are a postdoc, try to co-supervise or supervise PhD students. So always try to show that you have dealt with managing people, because the worst thing that can happen is somebody hires a PI and then the PI makes a really bad job of managing the people that they hire, because then you create problems for the department. You're not solving any, or you're not adding anything. So that is something that is, strangely enough, also looked at quite in some detail. So these are, I would say, the key things. And I think Lynn's point was really, really well met, is if you don't like writing, please do not consider a career in academia, because a career in academia consists of writing about your ideas and your accomplishments. That's all we do all day long.
Thank you very much. For the second question for our panel discussion today is, given how instrument dependent we are in our field, how do you start a conversation about getting or upgrading instrumentation facilities when you start your own lab?
So I guess I'll go again if
yeah, that's fine.
Yeah, so yeah, well, this is critical, right? If you're an experimentalist and basically your whole research program is going to rely on having, daily access to a mass spectrometer, then this is absolutely something you need to get right from the start. And there are different models. Maybe, this can be included in a startup, as mentioned, which is what happened when I arrived here. Maybe there's already proteomics groups or mass spectrometry groups in your department, and maybe they would promise you some access or some percentage of this. Probably this is kind of less ideal, but potentially still viable in some cases. Or maybe they would try to convince you that there's a core facility and you should access this one. This would probably be the worst situation, but potentially still viable. I don't know. I'm actually not sure. I'm aware that people operating under this model. But whatever it is, you need to make sure that it's right, and that it's right at the start, and that you're not in the situation of them saying like, well, we're going to recruit you, but we'll worry about that stuff later on. You know, you can write a grant when you arrive or something like this. This is definitely not going to work. So I think you need to have this agreed and written down before you kind of accept any offer. And I think you know, hiring committees will be sympathetic to some extent if you can make the case, or let's say head of departments or whoever is allocating the resources. If you can make the case that you absolutely need this, and then it doesn't make any sense for them to hire you without this, then I think people are, you know, will be reasonable. But I think it needs to be dealt with. And I think you need to be kind of strong in it. You need to sort of stand up to people to some extent. Like when I was coming here to Belfast, they did try to convince me to go for a shared instrument kind of model with a different group. And I kind of said, no, it's not going to work. And eventually they backed down. But of course they may not have, but in this case they did. Yeah, as far as upgrading instrumentation, I mean, this is a different story, and I think there's a lot less guarantees, and you have to probably deal with this down the road. Usually there's some institutional way to try to apply for these funds, but this can be hit and miss, and you need to deal with this as it comes. I would say worry about the first sort of 5 years when you're thinking about what position to take.
Yeah, so I think I totally agree with Ben, especially as I wanted to follow up on the needs. So when you start the conversation about like what instruments to get or whether you can get your own, is you need to know that the institution hires you and they want you to be successful. And when they create this job opening of hiring a mass spec people, they should have some expectations that this person need an instrument, either a shared one or their own. So when I think this is something you can start thinking even before the first interview. So if you talk to some vendor, the salespeople like Thermo, Bruker, Waters, talk to them and get some quotes so that you have a bulk number in your mind about like how much a mass spec costs. I mean, we always use mass spec, but when I was a PhD and postdoc, I never know how much it actually, I know it's expensive, but I know exactly how expensive it is until I actually talk to the vendor and salesman and getting some quotes. Then when you're having this conversation during your first interview, you can actually, you know what you need, and also I think because it's a conversation, it's a negotiation. I think negotiation happens in the first interview. It doesn't only happens after you get an offer. So because it's a negotiation conversation, you wanted to figure out the bottom lines for both parties. So what is your bottom line and what is the bottom line of the institution? So the bottom line of the institution would be depending on like, will they be willing to buy you an instrument or give you how much shared instruments, or give you free access to the instrument? And that you can figure out during your first interview, because you're going to go to see the different labs and talk to different people. Those are something probably easier to figure out with some facility people or the professors instead of the chair, because the chair might be, you know, sometimes they hesitant to commit on certain things. But a lot of the professors, especially assistant professors, they like to share a lot of the information with you, and you can chat with during your first interview. You can get some information about what kind of the bottom line of the institution is. And the bottom line for yourself would be, what kind of instruments do you need? And that's based on the needs of your research. So when you're talking to the institution, you don't want to start with, I want this instrument. You want to start with, what kind of research I want to do. And because I need to do this research, I need this instrument. And what is your bottom line? So obviously you can't ask for, I think it's very hard to ask for a $2 million machine when you start. So what is your bottom line? So as for me, my bottom line is I absolutely need my mass spec, but I don't have to get the super good ones. So I actually get a refurbished one, which costs much less, but it also works. And I negotiated with the instrument company to have longer longer period of warranty. So then that really helps. So I think knowing the bottom line of two parties and starting from like what you really need in terms of research will help with the conversation.
Thank you. And Leonard, do you want to add something to that?
Yeah, just briefly, because I don't buy mass spectrometers. I just buy servers, and they are very cheap compared to the prices of a mass spec. So most of my money is in people. However, one thing I do know about this whole purchasing and wider admin thing is that a key thing to never forget is that the university runs on a lot of, or any research institute, in fact, runs on a lot of administrative people. And Lynn already said it's important that you can talk to the right people about these kind of things. And knowing about costs and knowing about balancing your budgets and knowing about things like, do I pay a VAT or do I not pay a VAT, and which budget goes where, and how, and can I use this budget to buy that kind of thing? And all of these kind of things will come through your admin. So your best friends in the world are the admin people. And the admin people have an enormous amount of power behind the scenes because they make all the decisions. So always, always, always be very, very friendly with all the admin people. Also be very, very friendly with all the technical people. Also be very, very friendly with all the IT people, because they will hook your instrument up to the network. And whenever there's restrictions and you need to bypass these restrictions, it really helps if you bought a piece of chocolate for that nice man of IT, because he will go and you will go the extra mile. And it sounds very ridiculous, but this kind of stuff I managed to get done just by being extremely nice to these people that most people are not so nice to. That definitely helps. So it's a really silly piece of advice, but it will get you somewhere. Trust me, one day you will thank me.
Thank you very much. Just before I move on to the final question, if anybody has any question from the audience, please feel free to raise your hands or type in the chat, and we will address them after this question. For the final one, how do you decide the parameters of starting your own lab? That is the location of the university or the institute, the facilities and scope of research. How do you choose those?
Yeah, I mean, this is in a way a difficult question because to some extent it will depend a lot on the individual. I think for sure what matters a lot is the environment. So what does the environment mean? Well, for me, it kind of meant to some extent, you know, what we discussed already, you know, resources and available capability to have access to instrumentation and facility. But I also think it relates to what are other groups doing around you. And in my particular case, you know, what I'm interested in mostly is having good biology groups that I can have collaborations with. And so these were I guess high on my list. These are kind of the professional considerations. But there are also a lot of personal considerations. And so, you know, when I was leaving Zurich and trying to look for the next position, I did actually really restrict my search space a lot to mostly UK and Ireland, because I had spent, you know, 9 years in Switzerland, and I sort of felt like it was time to head in the direction of home, mainly for kind of personal and family reasons. So I took the decision to restrict my search space based on that. And this for sure dramatically, you know, restricted the opportunities that were available to me. And then everyone has to kind of make their own decision, you know, how much they're willing to sacrifice on let's say the personal or family side for the best opportunities. And as it happens, I think it worked out reasonably well for me, but I think this is always a big consideration. So yeah, let's say research environment and personal considerations, I think is what I would put at the top of this list.
Yeah, so I'll add on the, I think I talked a little bit about think about whether your lab is going to be a graduate student heavy lab or a postdoc heavy lab. So I grew up in a graduate heavy lab in PhD during my PhD, but during my postdoc, it's a postdoc heavy lab. It's very, very different culture. The postdocs are kind of doing their own thing, and then they can be more independent. We're graduate student, and also I have several undergraduate students in my lab. They need a lot of involvement and mentoring. So when you are thinking about, I got this piece of advice when I just I started my lab from another senior professor. He said that you need to have both the long-term plan that you're going to grow your lab into a unique program that's different from others, you also need to have some housekeeping project. So that making sure that you continue publish and being productive. So I've known I've seen some people don't publish at all for the first 3-4 years, and they publish a Nature paper, which is obviously super good, but there's the risk in there. And I think if you are a graduate student heavy lab, it's going to be very challenging, because the students I think still now, my most senior students in the lab is starting their 4th year. Most of the students, if they don't make mistakes, I'm already very happy. So I think there's times that you have the considers. So we are very, like, when we are starting our own lab, we're very mature in terms of doing research projects. But how do you kind of translate that into the productivity of your trainee is you really needs to be patient, and things take much longer than you expect. But it's like that. It's always like that. So and then they need time to grow. So when I first started, I had to start with some kind of low-hanging fruits, what we call, to keep our lab publishing. It also relieves a lot of my pressure and stress, and now relieves a lot of stress of the students. And it also helps to get early on grants, because they like to see some preliminary data. So those are the things you wanted to consider. Also, I think it's based on environment, and it's also based on personality.
Thank you. And Leonard, final one.
Yeah, I was relatively pragmatic, not to say opportunistic. By the time I was at EBI as a Bright Group Coordinator, because I really had this idea I want to be a PI somewhere else. Because at Amble you have this 9-year rule where after 9 years you're supposed to move on, which is kind of meant to seed people into the research environment worldwide, but especially in Europe. So I was applying for a few positions, and I actually got very close to working in Norway, because I got a job offer there, and it was only at the 11th hour, essentially, that my job application in Ghent, which I also had done, came through. And then, because I had the choice, I did what Ben did, and I went for the Ghent opportunity, because I had a family by that time. So I started my family in the UK, and I thought, well, it's better to be there. Plus, the Ghent position was a permanent position straight away, whereas the one in Norway was one where I would have to fight again after 5 years. And so, you know, all things considered, it was nicer to move there for family and career reasons, probably. But fundamentally, you really have to make the choices that have been elaborated by Ben and Lynn, which is essentially, do you care enough about the family thing to restrict your search and potentially not get an opportunity? And this whole postdoc heavy versus PhD heavy, I think that's a very important distinction to make. And it goes with the teaching as well, as Lynn said in her presentation. So I really like teaching. So I prefer to go to a university where I can teach, because I just think this is fun. It's also a great place to get students. So there are master students there. They can do a thesis in my group if they like me, and that means that I can then hire them as a PhD student potentially. So you get to train up your own talent. So that is a big advantage. In a research institution, you're always dependent on getting talent from outside coming in. The problem there is, if you're not sufficiently well known, it might be a struggle at the beginning to get them in. So these things you really have to carefully weigh, and these are the kind of things that are non-trivial to understand. One thing that can help you with this is by asking around. So if you're applying for a position, maybe get in touch with somebody you know who works there and ask them about what they found challenging or interesting when they started. These kind of things are actually very useful to find out, because the last time you want to, or the last point in your career you want to find out, is after you've accepted the position, you're there, and then you suddenly realize these kind of things. So never be afraid to ask people about that. In fact, if I were on a committee and somebody would say, can you tell me a little bit about starting your own lab, what it was for you, what it was like for you, and what you found challenging and difficult when they ask you if you have any questions, I think that would be a plus point on your application, actually, if you in an interview would do that kind of thing. And then the other thing is, of course, what I tell all my students, what my supervisor used to tell me: apply as much and as often as you can. Very simply for the reason that the more doors you knock on, the higher the probability somebody will open a door. And you can always say no. So the other thing, of course, is apart from this opportunistic thing, is you actually learn. Every time you do an application, every time you go to an interview, you learn. And if you apply to a place where you don't think you will want to end up for whatever reason, these are nice interviews to go to because you're relaxed. You don't want this job per se. So you're going to that interview relatively mellow and relaxed, and you can learn how to do interviews well. So by the time that job comes along that you really, really, really want to go for, you have some experience and you can probably do a better job. So never be shy to apply everywhere you can, and learn the ropes, and then you can always say no.
Thank you very much, and thanks to all the panelists. We have a question here in the chat box on negotiating for an instrument in startup grant. If the options on table are for a new instrument and a shared instrument, and one opt for the former, will that lead to any trade-offs? If so, how do you navigate such a situation?
Yeah, maybe I can make a comment. I mean, I think this issue relates to what do you actually need. So if you are a very technically oriented person and you need access to the instrument every day and you're doing these kind of technical optimization experiments and you spend a lot of time doing that, then I think you can make a good justification that you would need an instrument for your own lab, and you know, the people who are providing the resources will want to see that justification and that it makes sense. On the other hand, maybe if you have a more biologically oriented lab that needs more occasional access to mass spectrometry or to proteomics, then maybe having some kind of shared instrument situation or other access model is sort of okay. And so it really depends on what you need. And I think that the point Ling made, that the people who are doing the hiring and providing the resources, you know, have some expectations that they probably will have to provide some money. If they want to hire an expert mass spectrometrist, they will have to sort of provide the resources. So I guess if it's going to be this more shared model, then I think you would want it very clear how that model will operate, you know, and if you're, for example, sharing an instrument with another group, maybe it would be worth having a discussion with the other person in that other group before you sign your name on anything. I mean, the trade-offs are obviously that you save money by sharing resources. But of course, if you have an instrument for your group, then you have much more control. So for me, it was important in my case to really push to try and get this exclusive access to an instrument. But in other situations, it may not be the most critical thing. I don't know if I answered the question, but I.
Yeah, I think Ben made it pretty clear. We can see if other people have other questions.
Yep, please feel free to raise your hand. I think Matthew has just raised his hand.
Oh, one thing I wanted to add is when you are starting your lab and started purchasing, if you can get your own credit card, like institutions usually have some P card or credit card, get them. So although you have to do, so I have to do my own expense report because I use the credit card, but it's so much easier because if you rely on the admin to purchase all of your stuff, it's gonna be so slow. So the credit card usually has, like, for us is $3,500 something limitations. So, but most of the things are below, like the regular things are, you can use that. Yeah. So get your credit card up.
Thank you. Matthew, did you have a question?
Yes, thank you. This has been very interesting. I'm going to ask just a quick one. When you started, did your institution assign a mentor to you, or did you actually sought after getting one mentor that would help you throughout the process? Was that useful?
Yeah, so maybe I can start with that. So I did not get a mentor assigned back in the day, but nowadays Ghent University has a starting professor mentorship program, which is wildly successful, I would say, because a lot of people apply to that and there's a lot of people willing to be a mentor. In fact, I've been a mentor to several starting professors meanwhile, and it is a wonderful experience. It's both a wonderful experience to be the mentor, but also to be the mentee, I guess. And I would definitely recommend that when you start a new job in an institute, find somebody you like, take your time to find somebody you like, and then very politely ask them to be your mentor. They will likely be honored and will happily do that. There's 3 good reasons to do that. 1, you get somebody to share their experiences. 2, you get somebody to help you navigate your new job and the ins and outs of that. So whom do you talk to about what? What do you pay attention to for any kind of internal competition for funding or for advancement or what have you? And then the third thing is it will help you grow your network internally. So definitely try and get somebody to mentor you. And you don't have to do much. What we do in Ghent University in this program is every 2 or 3 months, we have a lunch. And then if 2 people really like each other as mentee and mentor, and the link is good, then that can be more frequent even. So you can have every month, but typically the agendas are so busy, it's every 2 months, and you carry that on for a year. That's usually enough. But definitely, Mathieu, that is a fantastic suggestion, and I would recommend anyone to do that. And in fact, I would say don't wait until you're a PI. You can also do this at the postdoc level, which we also have at Ghent University, and that works really well as well. The only downside is it's slightly harder to find people and to get people, but you know, you guys know and everybody else does not know, so go for it. Find somebody to give you some advice. It's very cheap and it's very easy.
Yeah, I fully agree with this. I mean, having local knowledge when you're coming in from the outside is invaluable. And particularly for me, it was the funding systems in the UK were very cryptic compared to ones I was used to. And there's a lot of specifics. And there's also just generally a lot of secret stuff in universities that you don't know how to do, but other people know how to do. So yeah, I fully agree with Leonard's comments.
Great, thank you. Well, we're slightly over time, so I think I'll wrap up and say thank you to Ben, Ling and Leonard for joining us. We hope that you guys got a lot out of this, and as Leonard said, feel free to join the Hooper ECR. Just drop us an email or send us a DM on Twitter, and keep an eye out for the next online panel discussion, which will be in a couple of months. So thank you again for joining us, and thanks Ben, Ling and Leonard. Thank you so much.
Thank you very much.
Thank you so much. See you guys in Hupo.